Now look at this.
What's up, about pain? We're going to do topic 6.1 on DNA and RNA structure. So what is DNA? DNA is deoxyribonucleic acid. The nucleotide, or the monomer of it, is going to be made up of a pentose sugar, being deoxyribose. It's going to have a nitrogenous base, so adenine, thymine, cytosine, or guanine, and it's going to have a phosphate group. RNA is almost the exact same, except for instead of deoxyribose, the sugar is going to be a ribose, and instead of thymine for one of its nitrogenous bases, it's going to have a uracil. DNA will be stored in the nucleus of a eukaryote and the nucleoid of a prokaryote. Eukaryotes and prokaryotes have a lot of differences in their DNA. So a eukaryote is going to have multiple linear strands, while a prokaryote is going to have a single circular DNA strand. Both of them will have something called a plasmid. Plasmids are going to be small extranuclear DNA. So it's just like extra DNA that's going to be within the cell. These plasmids have a lot of different genes on them. Okay, one gene can be providing assistance during replication. Another gene can provide resistance. So if there's resistant bacteria, it can pass that plasmid to another bacteria to allow that bacteria to be resistant. And a lot of scientists have used these plasmids to insert certain genes of interest into organisms. So the big example I can think of is that we have transformed bacteria to get it to make insulin. So those individuals who don't have insulin or aren't able to make insulin, we can now provide them insulin because we've, in a lab, transformed bacteria to make insulin. That was too long, I'm sorry, y'all. So DNA is usually double-stranded. And in the case of its double-strandedness, you're going to see that adenine is going to pair up with a thymine, and cytosine will pair up with a guanine. Notice that the adenine to thymine has 2 hydrogen bonds, and the cytosine to guanine has 3 hydrogen bonds. The way I remember that is that cytosine is the 3rd letter of the alphabet, so you can remember that 1, 2, 3, there's 3 bonds. Notice also their structure. So adenine and guanine have a double ring structure. This is called a purine, while cytosine and thymine are going to have a single ring structure. These are pyrimidines. Notice that there's always a purine and a pyrimidine bonded together. Why? Because it allows there to be a uniform width to the DNA. If you had 2 purines bonded together, the width would be too wide. If there's 2 pyrimidines bonded together, it would be too narrow. So by having a purine and pyrimidine together, it makes it a uniform width. So since we know things that are bonded together, let's do a little bit of math. So if there's 20% adenine inside the DNA, how much guanine is there? Well, there would be 30% guanine, because if there's 20% adenine, there's 20% thymine, which means 40%. There's only 60% left over. Half of it has to be guanine, so 30% is guanine. Remember, I'm doing this for every single unit, so hit that follow button, and AP Bio Pain is just us. Bye, y'all.